So cellular respiration is divided into three types. The first step, that is glycolysis, second TCA cycle, and third one, that is ETS or electron transport chain. So what is glycolysis? As the name suggests, glycolysis, glyco means glucose, or you can say sweet, sugar. Lysis, on the other hand, lysis means breakdown. So the breakdown of glucose called as a glycolysis. So glycolysis is a process through which a glucose molecule breaks down into 3-carbon compound pyruvate in presence of certain enzyme to generate ATP and NADH. Glycolysis occurs inside the cytoplasm. On the other hand, the TCA cycle and the ETS occur inside the mitochondria. Now, glycolysis is oxygen independent process. TCA cycle and ETS depends on oxygen. Now, the glycolysis is divided into two phases. So first one, that is preparatory phase or preparatory phase also called, and the second one, that is payoff phase. So in this video we discuss about the preparatory phase, and in the later videos we discuss about the payoff phase of the glycolysis. So preparatory phase of the glycolysis consists of several types of enzymes and several types of product. So first, that is glucose enter inside the cell, and the first step, that is the phosphorylation of glucose. So glucose is phosphorylated to glucose 6-phosphate in presence of an enzyme that is called hexokinase. It is a energy dependent step. 1 ATP is needed for the conversion of the glucose to glucose 6-phosphate. Mg2 plus is also needed as a substrate for the enzyme hexokinase. Now, glucose, there is a tendency of the movement of the glucose from outside, from inside to outside of the cell. So cell have to phosphorylate glucose because the negative charge phosphate form of the glucose does not move out from the cell, and it remain inside the cell. So because cell have to conserve the glucose. So the first step of the glycolysis of the preparatory phase is the phosphorylation of glucose. So glucose is transformed into glucose 6-phosphate in the presence of an enzyme hexokinase. ATP is needed for this, 1 ATP is needed for this reaction, and Mg2 plus is also needed to complete this reaction. Because Mg2 plus, Mg2 plus, the mask the negative charge of the ATP, and the actual substrate of the hexokinase is not ATP, is Mg ATP2 minus. So it is easier for the interactions of the gamma phosphate of ATP with the glucose. So this is the process by which glucose 6-phosphate is formed. Now the second step is the glucose 6-phosphate is transformed into fructose 6-phosphate in presence of another enzyme that is called as phosphogluco isomerase. Phosphogluco isomerase, or you can say phosphohexose isomerase. And Mg2 plus is needed. This is a reversible reaction. The third step of the preparatory phase of the glycolysis is very important. that is conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. So it's also one ATP is also needed in this conversion process, and the gamma phosphate is transformed to the fructose 6-phosphate to form fructose 1,6 bisphosphate. Mg2 plus is also needed for this reaction. Now this reaction is catalyzed by an important enzyme that is called as phosphofructokinase 1. The detail of phosphofructokinase 1 we already discussed in the earlier video, so please check out the earlier video, so you can easily understand what is the role of phosphofructokinase and what are the allosteric activator and inhibitors of the phosphofructokinase, because it is very important enzyme in the regulation of the glycolysis process. So the third step of the glycolysis is the regulatory step, is also a regulatory step of the glycolysis. So in this step, the fructose 6-phosphate is transformed into fructose 1,6 bisphosphate in presence of an enzyme phosphofructokinase 1, ATP and Mg2 plus. It is a irreversible step of the glycolysis. In case of plant, the name of the enzyme phosphofructokinase, it transfers the ATP from inorganic compound, inorganic phosphate, to the fructose 6-phosphate to form fructose 1,6 bisphosphate. Now the fourth step of the glycolysis is the fructose 1,6 bisphosphate is cleaved, or you can say transformed into three carbon compounds like dihydroxyacetone phosphate and glyceraldehyde. So glyceraldehyde 3-phosphate and glyceraldehyde 3-phosphate and glyceraldehyde so glyceraldehyde 3-phosphate and dihydroxyacetone phosphate is formed from fructose 1,6 bisphosphate. Now aldolase is an important enzyme, and so this reaction is catalyzed by the enzyme aldolase. Now fifth step of the glycolysis, the fourth step is also a reversible step. The fifth step is also a reversible step as a 4 and 2. So the fifth step is a reversible step, and in this step the dihydroxyacetone phosphate is transformed into glyceraldehyde 3-phosphate. This reaction is catalyzed by an enzyme called Triose phosphate isomerase. So dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate in presence of an enzyme Triose phosphate isomerase. So this is the last step of the preparatory phase of the glycolysis, and after that the glyceraldehyde 3-phosphate enter into the payoff phase of the glycolysis. So the first step, that is glucose to glucose 6-phosphate, is the first priming step of the glycolysis because ATP is needed in this step. And in the third step, that is the fructose 6-phosphate is transformed into fructose 1,6 bisphosphate in presence of ATP. That is why it is a second priming step of the glycolysis. And so in the preparatory phase, the 2 ATP is consumed. On the other hand, when we discuss about the payoff phase, we understand that 4 ATP is formed and 2 NADH formed in the payoff phase. So this is the steps of the glycolysis. Now what is meant by fructose 1,6 bisphosphate? Why it is called bisphosphate, not diphosphate? Because bisphosphate is called when the 2 phosphate is present in different position. So that is why it is called fructose bisphosphate. When we understand the structures of the fructose 1,6-bisphosphate, so we can easily understand that the two phosphate remain in two different positions. That is why it is called bisphosphate. If two phosphate situated in a same and attached with each other, it is called diphosphate.